Assalamualaikum. This is Ismat Farjana, Senior Lecturer, Department of Biology, Cambridge School and College, Dhaka. Plant absorb water through roots. It's very little bit difficult for understanding for the students. So I will try to, for better understanding this process, our students will did a practical class. This is very easy methods for better understanding. I think this is very easy for all of the biology students. Thank you.
Hello everyone and welcome to the session that we're going to be having today. Today we're going to be having a brief discussion on a topic that we've chosen, which is absorption of water and nutrients in plants. Even though it's supposed to be a really hard topic, our teachers have made it easy enough for us to understand and explain it in an even easier way. A group from our class will be showing a practical experiment on this topic alongside the discussions and descriptions that we've arranged. And I, Adiba of Class 9 English version, Cambridge School and College, will be hosting as it goes on. We'll start off by gathering some information on the topic that we're going to be looking into, which is the absorption in plants, as I said. So, the absorption in plants is one of those aspects which is really important for the well-living and the proper nutrition of the plants. The roots play the main part over here, which is absorbing the water and nutrients from the soil. The roots pass it to the upper body, and then the nutrients reach this vessel, which we know as the vascular bundle, the xylem and phloem tissues. They carry it to the parts of the plant where photosynthesis occurs, and we definitely know what photosynthesis has an impact. What's the impact of it? So that was a short talk on the topic that we're going to be looking into. Now we're going to look into the practical that our group has to present.
Hello, I'm Atika. Before starting the practical, I will just show you the things that are needed for the experiment. We will be needing 4 beakers, some fresh peperomia plants, some blue vitriol or ink, and a marker, and some oil. So my friends will now start the practical.
Assalamu alaikum. I am Marjuk Mahir. Today I will start the practical. This practical is a bit difficult, but it's quite easy for us. So we will try our best to make it easier for you. So I will be less talking and starting the practical. You can see a conical flask in front of me filled with water. So I am marking the water level. It is nearly 3/4 of the water filled of conical flask. Now we will just seal the mouth of the conical flask and keep it aside for 24 hours. Now we just have to wait for next 24 hours, and my part ends here. The next part will be conducted by my friend Shonthi. Thank you.
Assalamualaikum, my name is Abinta Shondhi and I'm going to do the part B. First of all, we have to take a conical flask. Then we have to put a 3/4 part of water in this flask. Then I'm marking the water level. Then we have to put oil in this water. Then we have to put a plant with root and keep it for 24 hours. This is the end of my experiment, and this is going to Ogni.
Hello, I'm Ogni. Now I'm going to be showing the part C. First we have a conical flask here. Now I'm going to fill it with 3/4 of water. And now I'm going to put a blue vitriol in it. And I'm going to mix it. Now I'm going to put some oil in it. And a fresh peperomia plant with roots. Now I'm going to keep it aside for 24 hours. My part is over. Now my friend Zubran gonna tell you the part D.
My name is Jubran. I will show the part D. First, take a conical flask and pour some water into the conical flask. Fill the flask with 1/4 part of the water. Then mark the Mark the water level. Then keep a plant into the conical flask and wait for 24 hours and let's see the observation. Now my part has ended. Now my friend Alif will do the observation part and show us what is the result.
Moving to beaker C, we can see that the water level has also been decreased and the stem and leaves have turned into blue. And if we go next to this beaker, we can see the leaves and stems has also turned red because they have given red color in it. After moving to beaker D, we can see that the beaker is quite empty because there was insufficient water in the time of practical. Now this is all I have observed throughout the experiment. Do you guys have any question?
I'm Maria and my question is, why was peppermint plant chosen for this experiment?
Because this plant is more transparent than other plants. As you can see, we have done our practical yesterday's class. You can see it's absorbs water through roots, so we can see where the water has gone. So that's why we have chosen peppermint plant than other plants.
Hello, I am Raha, and my question is, why did the water level in the first container remain unchanged?
We know that plant absorb water. You can see in my conical flask there is no plant inserted on it, so the water level remain the same. I hope you got your answer. Thank you.
My question is that why water level decreases in second, third, fourth container?
We can see that in the B, C, D conical flags there is plant, and plants absorb water through their roots. That's why the water in the conical flags decreased.
My question is that how the water reaches to the stem and leaves.
Now me and my friend will explain the full process through a drawing picture. Now we will start the drawing picture explanation.
As we know, plants absorb capillary water and other minerals through their roots, as this is shown in the picture. Plants absorb water and other minerals into the roots and go to cortex through epidermis.
From the cortex tissue, the water into the endodermis, and from endodermis, the water is transported to the vessel. From vessel, the water is transported to the stem through xylem tissue, and from xylem tissue, the water is transported to the leaves through the phloem tissue. And the full process of observation through root is done by osmosis and transportation process. My friend Tasin.
Hi, I am Tasin.
And Arafat.
Hello, my name is Arafat.
Helped us in the work of this observation of water through roots in doing this project work.
Well, thanks a lot to all my friends and teachers for their kind efforts. That's the end for this session, and hopefully all our efforts and the learning outcome will be helpful enough for all the other biology students out there. That's it for today. Thank you so much.